So this week we're going to be talking about the electrical properties of neurons. These remarkable cells, these neurons, these wire-like neurons, some with exceptionally long wires, right, that you could, make in pipe cleaners. Note that this one, even though it seems quite long, it's not as long as some of the longest wires that we actually have, axonal wires that we've got. But I want us to consider this week the nature of that electrical transmission. One of the most important things to consider this week is how neurons are set up to carry electrical messages, and that requires a great deal of energy input and energy expenditure to keep them in a state so that, should the right conditions arise, should there be a reason for sending an electrical message, the forces that drive the movement of charged particles that will carry the currents will be arrayed as such as to drive them, and generate, an electrical current that we call, actually, we'll see, an action potential. So neurons are kind of expending a lot of energy at all times. All these 86 billion cells that you've got are utilizing ATP. They are, distributing ions, which are going to be the charge carriers in our neurons, across membranes, so that given the right circumstances, we can initiate, the movement of these charged particles back and forth across the membrane, and that movement, those currents, we're going to see that go in and that go out, that in, out can propagate from one end of a neuron to another. So this week we're going to be talking about what's known as the resting potential. And the resting potential is, the neuron set up and ready to go. No actual currents, no long-range propagating currents are actually flowing. When the neuron is at its resting potential. But we're going to talk about how under the right circumstances, the neuron can shift and allow for what's called an action potential. This propagating current from one end of the neuron to the other, one end of the axon to the other. And I want to set you up, and I'll continue to point this out in our Zoom classes and other opportunities we get to talk. That the neuron, the terminology around the electrical properties of neurons actually derives from the original experimental investigation of neurons. So, in order to really examine the charge differences across the membranes of neurons, we needed actually early in the 20th century, into the mid 20th century, like a model organism that actually had neurons that were significantly large enough to allow for, us to place an electrode, right, on the inside of the axon, and then another electrode on the outside, so that you could connect them by wires and see if current flowed. So we actually called these electrodes potentials. So you could stick a potential on the inside of an axon, so this would break through the phospholipid bilayer and stick on the inside, then another potential on the outside. And in a neuron that was not actively sending currents, there was no action potential, there would be no action between the potentials. There would be a charge difference that was maintained when you compared the electrode or the potential on the inside with the one on the outside of the membrane, but that charge difference rested. So that was called a potential difference. That's the resting potential. And that's a neuron. A resting potential is a neuron set up and ready to fire. We're going to see it's very energy intensive. A lot of ATP has got to be expended to maintain that difference in charge inside and out. So a neuron is ready to fire, when something meaningful needs to be transmitted. But if a neuron's going to actually send a current, send a message, start sending messages down, you're going to need some action between those potentials. The axon that these early scientists studied, by the way, belongs to an invertebrate species called the giant squid. Invertebrates don't have a bony, vertebrae or skull. So they actually, in order to speed up neuronal transmission, instead of like myelinating, adding, those myelinating glia, having them wrap around, there are these little myelin sections here to insulate and speed up transmission. they just grow enormous diameter neurons or axons here. And in fact, the giant squid axon can be visualized, can be seen with the naked eye. You don't even need a microscope for that. So that was a perfect model organism for examining the charge differences across membranes, how before a neuron actually is stimulated, it's the charge is, there's a difference in charge inside versus out, but it rests, it's a resting potential, and how under the right circumstances when the neuron is stimulated, you get action between the potentials. So that's what we're going to be talking about this week. We're going to be talking about the electrical properties of neurons, because all these remarkable cells transmit information along their axons, along their length, via electricity.